Well, thank you so much, Nigel, and it's a great pleasure to be with all of you, and thank you for taking time out of your busy day to be here. Um, Nigel, very early in my career, I began reading Nigel's papers. He was probably about 5 or 6 years ahead of me in pediatric training, and he was an inspiration then and continues to be. And actually, in preparation for this talk, I reread one of his papers written in 1995. But at any rate, I'm going to talk to you about extreme prematurity, risk, and resiliency, and I have no conflicts. So I have two broad objectives. One is to describe the extremely low gestational age newborn study, the ELGAN study, which Nigel just mentioned, and then talk a bit about ongoing research that we are doing in conjunction with the ECHO collaboration. I believe many of you are a part of that. Sort of two pieces to that. One are early life influences on long-term outcomes after extremely preterm birth. And then secondly, a little bit about mechanisms that could link early life influences to these long-term problems. So when I was in college, and I guess they still had this, they had Cliff Notes, and the Cliff Notes you could buy if you hadn't gone to class all semester and read the Cliff Notes quickly and at least get a C on the course. So this is the Cliff Notes summary for my talk. And it begins with the premise that individuals born prematurely experience a higher risk of many chronic health and developmental disorders. I'll show you some data on that. And then the conclusion, or broad conclusion, is that early life factors, including prenatal factors, contribute to that risk and to that resiliency. And finally, that epigenetic programming is a candidate mechanism linking early life influences to persisting health and developmental disorders. And this is a mechanism that I should say has not undergone the ordeal to say conclusively that it's operative, but it's something that's quite interesting, and I'll show you some data from the ELGAN study. This is a recent paper some of you may have seen comparing the mortality risk after age 15, adults in Scandinavia. The upper 5 rows are for individuals born at 23 to 33 weeks of gestation compared to those born at term. And as you see, they're at higher risk for mortality. The same is true at 34 to 36 weeks, and even at 37 to 38 weeks, which was a surprise to me. These are the same data, just broken out into groups with the gestational age indicated with the different colors. Men in the upper bars, women in the lower bars. But as you can see, the gap does widen from about age 30 or age 15 to about age 35. And in a commentary that accompanied that paper by Casey Crump, who has done a lot of work on the long-term outcome after preterm birth, he commented that similar studies to the one I just showed you are needed in diverse socioeconomic contexts where preterm birth is more common, such as the US, and also studies are needed in more recent birth cohorts. You'll recall from the earlier slide that these were individuals born between 1967, I believe, and 2000. He commented that mechanisms might involve persistent epigenetic changes, and I'll talk a little bit about that today. And then importantly, that interventions should be developed and then formally assessed among individuals born preterm to try to provide some solutions for this public health problem. Extremely preterm birth is defined as birth before 28 weeks of gestation. It affects less than 1% of US births, but the survival has been increasing very steadily. These are actually data on the left side for a 1 kilogram baby. who faced a mortality risk of 95% in 1960, and then a mortality risk of only 5% 40 years later. And then since 2000, this has continued, and now about a third of babies born at 22 weeks, the earliest viable gestational age, survive. And as this audience knows, as survival has increased, there's more interest in neurodevelopmental impairment because these impact the ability to live independently and have cost for the individual, the family, and society. So to address this public health problem, a mentor for Nigel and for me, Alan Leviton, designed with help from Nigel and Carl Kuban and I, the extremely low gestational age newborn study. And I did want to just go back in history about 50 years to give perhaps some of the younger scientists in the room a sense of how long ago the origins of this study lie. And when Alan, who is an adult neurologist, had completed his residency, he did a fellowship in the lab of Floyd Gillis. And one of the studies he completed was a case control study of neonatal brain white matter abnormality at autopsy, and found an association with bacteremia with an odds ratio of 34. And so that led to some laboratory experiments in which kittens were injected with endotoxin, a product of a bacterial cell wall, and this could produce white matter damage in the kittens. And that model then has been replicated in all of these species you see here, as well as non-human primates. So this lipopolysaccharide or endotoxin model has been used to work out a lot of the molecular details behind the clinical findings that I'm going to talk about now. So this is my view of the overarching hypothesis that Alan and others were using, and that is that perinatal infections and perhaps even non-infectious initiators of inflammation led to brain disorders, and that some mediators then could be inflammation, and inflammation could be measured using measurements of these proteins you see listed here, like cytokines, chemokines, and adhesion molecules, and that one of the earliest manifestations would be white matter damage that again could be measured with neonatal cranial ultrasound. And this, of course, is very much in thanks to work that Nigel did at the beginning of his career. In addition, Alan postulated that there were protective factors. And these molecules you see listed here, neurotrophins and so forth, he postulated acted as endogenous protectors against the adverse influence of inflammation. And then even more recently, we've become interested in whether there's social factors that might serve as protectors. So the ELGAN study recruited 1,500 neonates between 2002 and 2004. The hospitals are shown here with red dots, East Lansing and Grand Rapids and William Beaumont. Chicago, we're in a hub led by Nigel. There was a North Carolina hub and then a New England hub. And using blood that was obtained when neonates were having blood taken for laboratory testing, and then a drop of blood was applied to filter paper and dried, using dried blood spots in the lab of Raina Ficharova, who I saw was on the call today, and I know is a collaborator of this team. In Raina's lab, 25 inflammation-related proteins were measured, including some inflammation markers. This is a partial list on the left column, and then the right column is a partial list of neurotrophic growth and angiogenic proteins. So in the first phase of the study, Neonatal brain ultrasound was obtained. Ventricular enlargement, also called ventriculomegaly, was a biomarker of white matter injury, as was echolucency. And then at 2 years, the children were evaluated with a structured neurological evaluation. The Bailey scales, which I believe many of you are familiar with, is a standardized test of early child development. And then a parent report measure of child problem behaviors, the CBCL, and an autism screen. And then in the second phase of study at 10 years, cognitive testing, academic achievement testing, and epilepsy evaluation. The CSI is another inventory for problem behaviors. And on a subsample, brain MRI. And then most recently in the ELGAN ECHO collaboration, which I'll elaborate on in a moment, 15-year outcomes with cognitive testing again. The mini kid is a structured psychiatric evaluation, and this time brain MRI was obtained on almost 70% of the sample. So the outcomes evaluated thus far have primarily been neurodevelopmental and mental health disorders, but through the ECHO collaboration, we've begun to collect data on obesity, asthma, positive health, blood pressure, and renal outcomes. And as this audience, I'm sure, appreciates, these outcomes not only are important in and of themselves, but probably interact with neurodevelopment. So a bit about the findings thus far. These blue bars are the rates of these conditions in the ELGAN cohort, and the red bars are the rates in the general US children population from CDC statistics for IQ under 70, cerebral palsy, epilepsy, autism, ADHD, asthma, and obesity. And as you see, only for obesity are the rates not higher for the ELGAN cohort, and of course, very strikingly so for cerebral palsy. There has been, unfortunately, sample attrition. 300 babies died in the neonatal intensive care unit, and then thereafter, there has been an average of about 3% attrition per year, unfortunately. So I'm going to turn now to just a brief summary of some of the key findings. Recall that inflammation was the exposure we were most interested in. And with regard to the relationship of inflammation to neurodevelopment, we found that multiple episodes of inflammation are more strongly associated with neurodevelopmental impairments than a single episode. Secondly, that multiple brain-related outcomes are associated with inflammation. And third, that correlates or initiators of inflammation or antecedents of inflammation include both neonatal factors as well as prenatal factors. And I understand that a number of the people involved with CHARM are on the maternal health side, and so I hope this will be of interest to you. These are coronal views of the brain with ultrasound showing pictures of cerebral white matter shown in the yellow. Just below the yellow circle is a very large ventricle, another manifestation of cerebral white matter injury. And then here's another more solitary echolucent area. And when we looked at the relationship between elevations of the proteins that I mentioned earlier, measured on dried blood spots in Raina's lab, and it has how they related to cerebral white matter injury, you'll see underlined a number of odds ratios that were in the range of 2 to 3, particularly for IL-8 or interleukin-8. And these protein elevations present on day 1, and day 7, or days 1, 7, and 14 were each associated with that outcome. So we did begin to think, and this is thanks to Olaf Donman, who obtained funding so that measurements could be made on the dried blood spots collected on days 21 and 28. And he realized that if a child had an elevation of one of these inflammation proteins on day 1, they were 2 to 4 times more likely than peers to have an elevation on day 7, 21, and 28, as if either inflammation was being sustained throughout those 28 days or was resolving and then recurring. We don't, from these measurements, know whether it's sustained or recurrent, but nonetheless, empirically, these multiple day elevations are more strongly associated with severe developmental delay as assessed with the Bailey scales than is a one-day elevation. And in this slide, the mental scale scores on the left, the motor scale on the right, and within those scores, the odds ratios for a one-day only elevation, as you see, are smaller than the odds ratios for a multiple day elevation. I showed you that slide was for cytokines, but the same thing applies for these other inflammation molecules that were measured. Somewhat perhaps corollary to that is that in this slide on the left are odds ratios for an antenatal inflammation in the placenta, measured histologically. And then in the middle are odds ratios for a postnatal inflammation in the neonate's blood. And then on the far right are odds ratios for elevations in both epochs of time. So this seems to be somewhat, I think, a corollary to that earlier finding of multiple day elevations. I mentioned earlier that inflammations seems to be associated with a range of brain-related outcomes, such as the white matter injury on ultrasound, delays on the Bailey scales, but also at 10 years, attention deficit hyperactivity, learning difficulties, intellectual deficit, and the social responsiveness deficit. And this last study was even among children who had normal IQs. I also alluded earlier to these neurotrophic or putative endogenous protectors. And this slide shows the joint relationship of elevations of neurotrophic proteins, elevations of inflammatory proteins, and cognitive impairment. So the height of the bar is the rate of cognitive impairment in 9 subgroups, and the 9 subgroups were defined according to how many neurotrophic protein elevations going from left to right, increasing numbers, and how many inflammatory protein elevations going from back to front, or front to back, increasing numbers. And as you see at the front right, with a rate of cognitive impairment of 10%, these were the children with more neurotrophic protein elevations and fewer inflammatory protein elevations. And in the back, 56% cognitive impairment. These are the children with low neurotrophic protein elevations and high inflammatory protein elevations. So to summarize what I've said so far, sustained or recurrent elevation, the multiple elevations of inflammation proteins are more strongly associated with neurodevelopmental impairments than are single-day elevations. Inflammation is associated with multiple brain outcomes, and then these proteins that Alan Leviton has referred to as endogenous protectors are also associated with risk, this time inversely with a lower risk. So that led to this question of what are the sources of inflammation, or what are the antecedents or correlates. And the pediatricians in the room will appreciate the importance of these conditions as potential correlates of inflammation, because they are so common among extremely preterm neonates. Retinopathy of prematurity, brain hemorrhage, necrotizing enterocolitis, and bronchopulmonary dysplasia. And in the ELGAN cohort, each of these four, in addition to sepsis, which I didn't have a picture for, are associated with increased levels of the protein biomarkers of inflammation that you've seen in earlier slides. So these are certainly correlates and antecedents of inflammation in the neonatal period. And they're also predictors of the risk of a low IQ. So in this slide, I've just plotted the number of those four morbidities that I just showed you against the percentage of children who had an IQ under 70. And you see there is a relationship here. And so it's natural then to ask the question, well, we have therapies to prevent those morbidities. How well have they been shown to decrease the risk of neurodevelopmental impairment? So I've listed here those morbidities in the left-hand column, and then in the middle column, some interventions that have been shown either by strong observational studies or multi-site randomized trials to decrease the risk of that morbidity. But as you see in the right-hand column, these have either not been studied in their relationship to neurodevelopmental impairment or been found not to be preventive. And so the picture to me seems more complicated than simply saying we can target these neonatal interventions and the problem is solved. And so not only do those therapies have small effects on later outcomes, but many children thrive despite having those morbidities. Nigel showed you the book, and on that cover is a little 22-week baby that I took care of that had many of these conditions like chronic lung disease. And I think you can see in the lower right corner, she's thriving quite well. And I think on her, I think I'm not supposed to tell you what her Bailey score is because that would be a HIPAA violation, but suffice it to say she did very well. So that's led then to our being interested in prenatal factors as an opportunity for having even larger impact and having an opportunity for an earlier impact. So to that end, Raina Ficharova looked at the relationship of placenta bacteria to neonatal inflammation, found an association, looked at socioeconomic disadvantage, and that also was associated with neonatal inflammation, looked at intrauterine growth restriction, and looked at pre-pregnancy obesity. And so these factors then seem to be related to neonatal inflammation. And when we looked at whether they were associated also with neurodevelopment, we find indeed that they are. So other prenatal factors that we now are looking at are smoking during pregnancy, prenatal, pre-pregnancy diabetes, maternal pre-pregnancy asthma, and environmental pollution, air pollution, which we can capture with geocoding, metals, which have been measured now in the archived umbilical cord specimens, and then organic chemicals that we are working on a plan to measure these using dried blood spots that were archived from the mothers. And as I mentioned, we're doing this work in collaboration with the environmental influences on child health outcomes. So this is a consortium of existing cohorts. We are charged with studying early life environmental exposures, their impact on pediatric conditions such as neurodevelopmental impairments, and we're charged with asking solution-oriented questions. And through this consortium, it also gives us an opportunity to study mechanisms. So I'm going to turn a bit from thinking about identifying risk factors to identifying mechanisms, because that might yield insights into how we could interrupt these links. So this is just a sort of a broad scheme of how could an early life exposure listed over on the left, how could that relate to an outcome in adolescence listed out on the right? Well, one way, and this is that top row, I don't know that I have a pointer, but maybe this is a pointer. Yeah, so one way would be that the early life exposure might disrupt organ development. So a large intraventricular hemorrhage, as I showed you a picture of, might be an example for that. And then that would then have lasting effects. Secondly, you could have a prenatal exposure like maternal smoking that continues postnatally. And that would appear to be a link between early life exposure and later outcome that would be in part, only part of the story, because that exposure is continuing postnatally. Then third, you might have inflammation, as we talked about with placenta bacteria causing inflammation, and that inflammation then has short-term effects, but maybe even lasting effects. And then finally, and something I'll spend some time talking about now, there's this possibility of programming biologic responses through epigenetic changes like CpG methylation and placenta. So if an early life exposure, let's say tobacco smoke, alters CpG methylation during a very susceptible window in the placenta, that might have effects on the fetal development, and it also might program for long-term altered biologic responses in the offspring. And so if that offspring encounters a triggering exposure way out here in childhood, that might further add to the damage. So you can see why it would be very important to sort this out, because it has implications for the strategies that would be preventive. And this idea of programming by maternal conditions, maternal exposures, leading to maladaptive responses that then lead to disease risk goes back to 1987 to David Barker, who sometimes this has been referred to as the Barker hypothesis, but more generally refers to the idea of developmental programming of the response to stressors throughout the lifespan. So here I've interposed between perinatal infection and non-infectious initiators, not only perinatal inflammation, which we've discussed earlier, but perhaps there's also early life programming of responses to stressors that continues through infancy and perhaps even through childhood, because one thing I forgot to mention is that these epigenetic marks may very well persist throughout the lifetime. It's not clear how readily those can be modified and what kind of interventions might modify them, but it's certainly plausible that they could persist for a long time. The actual molecular mechanisms for epigenetic control include non-coding microRNA, include histone modifications, and include DNA methylation. Most of the focus has been on the non-coding microRNA and DNA methylation. And so far, most of the studies I'll show you focus on DNA methylation. So I think many of you know this already, but I'll review for the rest of the group that DNA methylation can be thought of as a light switch for gene expression. So in this top figure, the cytosine guanine islands, these CpG repeats, is unmethylated, and the gene is then available for gene expression and and synthesis of protein. But in the lower bar, the CPGs are methylated and are not open for gene expression, and the protein then is not made. So this CPG methylation can be thought of as serving as a switch for the control of biological processes, and disruption of the CPG methylation then could disrupt epigenetic programming. So what kinds of things might alter the methylation in placenta? This is probably, well, I should say this is definitely the pathway that has been that has received most of the research focus. This is the HPA axis, and you see here the hypothalamus, which gets signals of a stressor in the environment. This could be Terra, for example, and it then produces corticotrophin releasing factor, stimulates the pituitary to make ACTH, which stimulates the adrenal gland to make glucocorticoids, and adrenaline, which is adaptive in the short run, but is maladaptive if the threshold for stimulating glucocorticoid synthesis is altered such that there's chronic exposure to heightened levels of glucocorticoids. As you all know, the placenta is a critical organ for fetal development, producing hormones critical for development, giving antibodies to fight infection, nutrients, but it also serves as a transporter of toxins. The maternal characteristics are associated with placental function, things like mother's weight, her nutrition, infections she encounters. And placental function has been tied to later life health, to fetal growth restriction, for example. In the ELGAN cohort, placental abnormalities have been associated with neurocognitive outcomes. So the first epigenetic question we asked within the ELGAN cohort was to try to probe the question of why are there sex differences in outcomes in the ELGAN cohort. So the blue bars are the rates of these conditions in girls. The red bars are the rates of these conditions in boys. The odds ratios here range from about 1.5 to 2, higher rates in boys. And so that's why we asked the question of, are there differences, sex differences in the placenta? This work was done by Elizabeth Martin in the lab of Rebecca Fry, the co-PI for our study. And there, as you know, the placenta is sexually dimorphic in a number of ways, such as energy metabolism, receptor expression. But we found within the archive placentas from the ELGAN cohort that it's also sexually dimorphic with regard to CPG methylation. So the far right bar shows CPG methylation differences in the XY chromosomes, no surprise there, but all the other dots that are above the red line are statistically significant genes that were differentially methylated in girls and boys within the ELGAN cohort. And these genes related to proteins in the immune response and also into transport genes. So it's quite plausible then that that differential synthesis of the proteins could in fact explain some of the differences in outcome. So this is the model then that we hypothesize that maybe these in utero environmental exposures are processed or transduced differently between the girls and the boys. That results then in different epigenetic programming and different disease susceptibility. In another epigenetic experiment, Martha Tomlinson who also worked in Rebecca's lab, followed up on the observation that certain microorganisms in the placenta are associated with neonatal inflammation. And so she looked at the CpG methylation in placenta in relationship to microorganisms. And just to briefly summarize, on the left are methylation patterns for placenta that contain lactobacillus. On the right, for methylation from streptococcus, placenta that contains streptococcus. And you can see rather striking differences. And along this line, lactobacillus in the placenta is associated with lower neonatal inflammation and with improvement in some of the neurodevelopmental outcomes at 10 years. So again, some plausibility that one of the mechanisms that contributes is this CpG methylation. And one of the pathways that was differentially methylated was in fact in the NF-kappa B, which is sort of a master switch for inflammation. In the interest of time, I'm going to skip this one. So I mentioned briefly earlier that socioeconomic status of the mother was associated with neonatal inflammation, and it's well known to this group that the socioeconomic status is associated with different outcomes. And so Hudson Santos looked at whether the DNA methylation in placentas was different for mothers who had varying levels of socioeconomic status. And he used as indicators of SES, maternal education, marital status, eligibility for Medicaid, and then eligibility for SNAP. And again, just to remind you that there's a very plausible link between the SES and these biological processes such as the HPA axis. So Hudson did find, in fact, that there were 35 CpG sites with increased methylation, 14 with decreased methylation, in association with one or more of those socioeconomic factor indicators. And then lastly, to sort of complete the loop, we've looked at whether CpG methylation of some of the HPA axis genes is associated with cognitive impairment in the ELGAN cohort. This just reminds us of the HPA axis that I just showed you, but also to show you that down at the bottom, you'll see BDNF, and you might recall from an earlier slide that that is in fact one of the neuroprotective factors that we looked at. Interestingly, BDNF also is involved in trophoblastic growth, which would be important in placental implantation. So how did those genes, are those genes associated with later life cognition in the ELGAN cohort? And you see here odds ratios that were significant. On the left are the gene names. And first, in this slide, I just highlighted genes that are related to glucocorticoid, glucocorticoid receptors, glucocorticoid synthesis. And then in this slide, I show BDNF genes. And again, recall that that's one of the neurotrophins that we looked at in ELGAN and is important in implantation. So just to wrap up then, where are we going from here? We have data on CpG methylation in placentas that were archived from the cohort. We now have messenger RNA expression in those placentas, non-coding RNA. We have GWAS, and then we have some data on environmental toxicants, and we can continue to pursue the avenues that I've been describing briefly about mechanisms linking early life influences to later life outcomes. So I just want to Highlight then the key findings. One is that neonatal inflammation is associated with prenatal factors as well as neonatal factors. This could be an opportunity for intervention. Secondly, inflammation is related to cognitive impairment, autism disorder, and reduced brain volume. And then secondly, that CpG methylation of target genes is associated with some of the exposures, such as placental microbes and socioeconomic status, and one of the outcomes, cognitive impairment, and that CpG methylation is sexually dimorphic. So to sort of harken back to the cliff notes, in addition to reducing neonatal morbidities, targeting prenatal factors could have more long-term benefits for neonates born extremely preterm, and mechanisms linking early life exposures to later life outcomes might include placental epigenetic changes and or prenatal programming of inflammatory or stress responses. These are the site investigators for the first phase, and the study, of course, could not have been accomplished without them. You'll see Padu Karna from Michigan State and some other people whom you might know, Dan Batten, Ed Beaumont, and then Michael Schreiber. This is one of the early investigators meeting. You'll see a young Nigel Panna there in the front. And in the on the right to the back is Alan Leviton, who created the ELGAN study. In the second phase, of course, Alan, Carl, and Nigel continued to be involved, but we recruited some developmental pediatricians and psychologists because now the children were 10 years of age. And then in this ECHO collaboration, the number of people involved has really mushroomed, which is just a joy to have these opportunities to collaborate with such a broad array of scientists. And then finally, the funding sources in the upper left is Rebecca Fry's lab. Rebecca is the co-PI with me of the ELGAN ECHO cohort, and Rebecca's team, many of the people you see in that photo worked on the projects that I showed you earlier. It's just been an amazing collaboration. So let me stop there and see what questions or comments, suggestions people have.
Anybody have any questions from Michael or comments?
Yeah, I have a question, Nigel.
Go ahead, Doug Rudin from Wayne.
Mike, this is Doug Rudin. I enjoyed your talk. I enjoyed yours and Rebecca's talk yesterday too. I thought I'd be bored today, but I thought they were nice and complimentary. Sort of.
Thank you for coming again, because it was a bit repetitive.
I checked the list of people yesterday, and I think I might be the only one who saw both. Someone can correct me if not. But one thing that, so there are two explanations for the methylation changes and the gene expression changes, you know, with like bacterial exposure or environmental exposure. Either the methylation changes in like a certain number, small percentage of cells, and you see that reflected in the data, or the number of cells change and the methylation doesn't really change. So what we're doing, like with Gilmore in mice, we did benzene studies and we did RNA-seq and methylation changes, and then you can do this technique called deconvolution or single cell analyses where you get the proportion of the different cell types. And for instance, in the benzene exposure, we found that the spongiotrophoblast layer of the placenta was shrunk. And when you do H&E staining, it's really obvious it's much smaller. It's like greater than 50% reduced. So have you tried doing that sort of thing with your data? Or if not, I mean, we can certainly help you with that sort of thing. Because I am part of your ELGAN 2 grant. So. Yes,
you are. Thank you. And I do remember, Doug, talking to you about maybe 2 years ago about this single cell deconvolution. The answer is we have not. We do whatever statistical methods are used to account for cellular heterogeneity. And you would know more than I how well those adjust for the change in cell type.
Yeah, no, I mean, the techniques are getting better, and there are multiple techniques available for doing that. So we have some people in my lab working on those techniques and trying to improve them. So you don't really need you don't need to do the single cell analyses yourself. You can take other people's single cell data and use that data to make a signature matrix and deconvolute your data.
That would be a wonderful collaboration.
Yeah, so we should talk more about that.
Yeah, because the grant that you're a part of, in fact, Doug, we want to look further into the sexual dimorphism. That's the main thrust of that.
Hi, Mike, this is Jane. I have a question for you. But first, I want to say what a fabulous presenter you are. You took such a comprehensive look and gave it to us in an easy to way understand. I can see how what Nigel said at the beginning, you're a good, have been recognized for your teaching, mentoring. I can see why. So thank you for all that. My question for you is about if you have spent much time looking at the infant feeding in terms of, how the formulas and the human milk fortifier have changed over the years, and people get different things in different NICUs, and then if you have any postpartum infant feeding data as well.
Yeah, that's a great question, Jean, and that's probably an opportunity maybe for a collaboration with you. We have experts in obesity, but we don't really have experts in nutrition on our team. To answer your question, we have published on growth rates while they're in the NICU. That was more of a descriptive study, and then and this is with Mandy Belfort, whom you might know of. But anyway, in collaboration with Mandy, we're looking at inflammation as it relates to the growth during the NICU stay. And she presented this last year at PAS. And her conclusion was that the babies who had higher inflammation seemed to have a bit of a inefficiency of utilization or absorption of their caloric intake, in that they didn't grow as well as would be expected, whether that's a change in their microbiome or absorption or even metabolism, she doesn't know. Or maybe the basal metabolic rate goes up in the face of inflammation. But that's one finding about growth in the NICU. The second thing we're in the midst of, and we proposed this for an echo-wide project. So if you're interested, we'd love to have your expertise on that paper. But we are looking at first and second year growth as predictors of later asthma, obesity, and neurodevelopmental outcomes in the preterm populations. Why are we limiting it to the preterm populations? Because as you know, when they're in the NICU, they tend to experience negative growth for a while. There's a very long recovery. So they tend to be discharged with weight Z-scores. It's not unusual for them to have a weight Z-score of -2 when they go home. There's definitely not a Gaussian distribution centered around 0 when they are at their due date. So over the next 2 years, then they tend to move towards higher Z-scores, and some of them, particularly nowadays, continue to overshoot. And so while they didn't at 10 years of age or at 15 have the rates of obesity in the general population, their rate of obesity is not trivial at all. It's in the range of 15%. And so we have this hypothesis that maybe that rapid change in Z-score in the first year is not advantageous. Traditionally, it's always been thought that rapid growth would help the brain grow faster, improve the neurodevelopmental outcome, but it might also lead to inflammation, right? If you overdo that and you have more visceral adiposity, and adipose tissue makes IL-6. And so that's sort of the theme, is that maybe there is a U-shaped relationship where very, very poor growth in the first year is probably not good, but maybe very rapid growth in the first 2 years is also not so good. And that would have clinical implications for, when you do follow-up of these NICU graduates. I don't know if I quite answered your question, but that's all we've done so far. We've not looked at, I think you asked about human milk fortifier versus human milk feeding versus formula. We haven't done much of that. We do, one limitation we have is we only have data through day 28 on that. So we have daily data 1 through 7, then data on day 14, 21, 28. But if you have ideas of how to utilize that data, we'd love to think about collaborations, because we don't have the nutrition expertise. I think
that is part of the beauty of ECHO, isn't it? You tap
Yes.
into all kinds of people. Well, thanks, Mike. Thank you so much.
Yeah.
I see that Kim McKee, before I get to pressure in a second, Kim McKee had a question in the chat box. Kim is at the University of Michigan, an epidemiologist, works with us. Michael, do we know anything about persistent inflammation into childhood?
Yeah, Kim, thank you so much for asking that. I didn't explain it very well, but just as there is a lot of literature, particularly preclinical models, but now even emerging studies in humans, that the HPA axis can be programmed for dysregulation, so that thereafter you have a exaggerated or low threshold adrenal corticosteroid release. Just as there can be dysregulation of that pathway that's programmed and then persist, there's emerging evidence that the immune response can also be programmed. And so there's this paper that's now 10 years old that talks about embedding of the inflammatory response. And I think that's what you're getting at, is these children who experienced initiators of inflammation early on, did that then lead to epigenetic marks that would cause them in the future, if they encounter air pollution or anything that incites inflammation, to have a heightened inflammatory response? And we did build that into the ELGAN ECHO grant as an exploratory aim, where we would take blood from the teenagers here at UNC only, because you have to have fresh blood, and then expose them to something like IL-1 beta or LPS, and see if there was any correlation in their early life DNA methylation signature and their response later on. So I don't know the answer, but it's a really fascinating question. It would have tremendous implications, I think, for how to help these children.
That would be one interesting experiment, Mike. Now, Prachi Shah, who is a developmental pediatrician at the University of Michigan, has a question.
And Dr. O'Shea, what a terrific talk you gave. Thank you so much. And I'm new to this group through the generosity of Nigel. So thank you, Nigel, for bringing me in. I was really interested in your findings looking at the social determinants of health and the CpG methylation, and specifically your SES paper. And I noticed that your construct for SES was basically sort of like a multifactorial construct. It had education and poverty and sort of like nutrition markers, Medicaid. And I wondered if those items were examined individually. So if, for example, was education the driving factor for the methylation changes, because that might allude to some intervention that might actually be actionable to change sort of methylation patterns and outcomes.
Yeah, that's a great question. And we, Bob Joseph, who is a developmental psychologist, did ask the same question. And he took our 10-year data. And so Medicaid eligibility, for example, we were thankfully not able to study because of Affordable Care Act. And so eligibility changed so dramatically. With marital status, it wasn't quite as, didn't move as much. But maternal education, we did have a significant number of mothers who had increased their years of education. And so Bob looked, controlling for mother's education at baseline, at the birth of the child, he looked at change in education or increase in education as another variable. And we didn't really have sufficient sample size to reach a statistically significant result, but the trend was clearly that also predicted better outcome. And so I do think that it's a great idea, and we need to do more of that. We now have the 15-year data, so we're beginning to do that over a longer span of time. We're somewhat limited by sample size, and so that kind of project would be ideal to utilize the ECHO-wide consortium, where we can probably double our sample size. So I hope you'll continue to stay interested, and through Nigel you could get involved in some of these analyses, because I think there's so much to learn about not only these questions about risk factors, but also just some of the
Protective factors.
Yeah, some of the resiliency protective factors. Just one other comment, and that is that we were very neonatology centric, medical centric when we started this study. And my regret, but also the opportunity to do it better next time, would be that we would broaden our look at risk factors to include some of the things that you're mentioning.
And sort of relatedly, were there measures of trauma, so sort of prenatal, perinatal trauma for the mothers, kind of like an ACE score, or something that could be a proxy for ACEs that might be sort of a contributing factor or something, or even a confounder to some of these?
Yeah, another great idea and a great question. And again, we were too medica-centric. We did at age 10, though, we did get an inventory of adverse life events like death of a parent and incarceration of a parent. So we're beginning to look at that data. And then through ECHO, of course, we're getting a much broader array, but it's retrospective, so it has some limitations. But again, that's another thing that if we design the study again, that would be the kind of thing we would need to collect a much richer data about.
It's a beautiful study, and I've learned so much. Thank you.
Yeah, thank you.
Well, it's past 2 o'clock, and I just want to thank Michael for giving us this wonderful talk. Also to point out to people around here that a lot of people around this audience have come to our Michigan, what we call our Michigan cohort march to do some analyses. But I think you hear Michael's open invitation to also link up to the ELGAN code, which has a representation, as pointed out, in our state, but is also beyond our state. And then, of course, that links to a lot of other preemie cohorts in ECHO. That is to say, not just the preemies who might be in any cohort, just because preemies are everywhere, but specifically focused on preemies such as Judy Ashner's cohorts and Mike's, and there are some others too. So if you have special questions, Prachi, about stress, these kinds of emotional stresses, emotional trauma, which are being well collected, I think, in ECHO right now, I think there's some wonderful opportunities to work, epigenetics for Doug, nutrition for Jean, trauma for Prachi, and so many of you. And yes, Reina, you're doing all the measurements for all of us and helping us understand everything about inflammation and other biomarkers. We continue to work with Reina Fichurova. So there's a lot of collaboration going on even as we speak. And so thanks everyone for attending, and keep in touch, and let's give a kind of a virtual.
Thank you all for the opportunity. It was a real pleasure being with you, and look forward to the future collaborations.
Great to hear from you, Michael. A real pleasure.
All right. Thanks.
If you have other questions, maybe write them to Michael.
I'll put my email. I'll put it in the chat here.
Yeah, Michael's going to tap his email into the chat for everyone. I'll keep this going for a little while longer.
And I'll share the slides in the next day or so.
Oh, wonderful, wonderful.
Thank you, Dr. O'Shea.
Yeah,
And
thank
thanks.
you.
Thank
Take
you so
care,
much.
everyone. See you soon.
Bye-bye.